It's a very random class I'm doing. Anyway, there's this photo that I put at the beginning of the video. That is a photo from USMLE First Aid Step 1 book, and it's a very nice flow chart because for people who do not have any concept in biochemistry, no basics, nothing for them, it's like a good starting point to get an overall idea ki how the cycles are linked with each other and what are the substrate level phosphorylation and where all the ATP is released, what are the rate limiting enzymes and all those small stuff, you know. So what I want you to do is that if you have two devices, two phones or like one phone and one tablet, then open that thing in one device, and in the other device you watch this video which I am making, right? And you can use that flowchart as a reference to make the diagram, like I'll modify it. I'm going to modify that flowchart slightly at only the required places, so you can use it as a reference, like overall this is what I'm going to teach. Anyway, so see here, it was a lot of struggle how to write in how much size letter so that everything will fit in properly. So anyway, I wrote it all down and now I'm going to teach you. Okay, so see this is how you have to write. So here you see, from glucose to glucose 6-phosphate, from somewhere that extra phosphate is coming. So here you can see 1 ATP is getting consumed. The enzyme is hexokinase. If you call it glucokinase, it is also okay, because glucokinase is a type of hexokinase. Hexokinase, which type? Type 4 is glucokinase. What else special about it? Listen, this is a reversible pathway, like you see here. From glycogen, if you want to form glucose, you can also come like that, na? So from glycogen you can break it down. First the 1,4 bond, alpha 1,4 bond will break, then the branching points will be there, like see, this is a glycogen. In glycogen there are many glucose molecules, so that is alpha 1,4 bond. But when there is a branch, then that is alpha 1,6 bond. So first of all there will be 1,4 bond breakage, then there will be 1,6 bond breakage, these points, then this is again 1,4. अभी branch के अंदर, within the branch, again it is alpha 1,4. So like that, finally you will get glucose 1-phosphate, and then it will become glucose 6-phosphate, and then finally glucose. So basically this is a reversible reaction. Okay. Now why I wanted to tell you is that from glucose 6-phosphate to glucose, that enzyme is what? Glucose 6-phosphatase. Glucose 6-phosphatase. That is, you have to break down glucose 6-phosphate. What will you get? You will get glucose. And if there is a deficiency of the enzyme glucose 6-phosphatase, then that disease will be known as what? Von Guericke disease. Von Guericke disease is what? Glucose 6-phosphatase deficiency, not the hexokinase. This is the reverse. Glucose 6-phosphatase deficiency. Got it? Now from glucose 6-phosphate you have fructose 6-phosphate. This is also reversible. It's okay, but this is just an isomer. It doesn't have much of an importance. But what is the importance exactly? The importance is that this glucose 6-phosphate is what traps the glucose inside the cell, because there is this transporter, right? There is this GLUT4. There is this GLUT4 transporter in normal cells that will trap the glucose, but GLUT4 is a bidirectional transporter. It will go from outside to inside and inside to outside, everything. So how to trap the glucose? So once this glucose comes inside, then it will be phosphorylated into glucose 6-phosphate. Now it is bidirectional, but it can transport glucose outside, but not glucose 6-phosphate. So now this is how this glucose gets trapped inside the cell. So glucose 6-phosphate, what is another point about it? The point is ki this traps the glucose inside the cell. Okay, anyway, then fructose 6-phosphate, no one cares about fructose 6-phosphate. And then from fructose 6-phosphate you have fructose 1,6-bisphosphate. 1,6-bisphosphate, that means there are two phosphates. So from somewhere the phosphate came. So here another ATP has been consumed. Okay, another ATP has been consumed. Now tell me one thing. What is the name of this enzyme? You don't need the name of all enzymes, but this is important. Why? Because this enzyme name is PFK1, phosphofructokinase. Phosphofructokinase 1. Why it is important and why I am telling 1? Because there is also a PFK2, phosphofructokinase 2. If there is PFK2, then what will be the product? For fructose 6-phosphate will become fructose 2,6-bisphosphate. This is if the enzyme is PFK2. Okay. So, okay, what else I want to tell you? Fructose 2,6-bisphosphate. Why the cell forms this? You don't need fructose 2,6-bisphosphate. Why will I form fructose 2,6-bisphosphate? That is because it is a kind of allosteric regulator. which basically means that if there is more of fructose 2,6-bisphosphate in the cell, then it means that glycolysis is not happening that much. That is why the fructose 6-phosphate is getting diverted towards the other side. So if there is more of fructose 2,6-bisphosphate in the cell, it will give a feedback. It will give a feedback that, hey, your glycolysis is not happening properly. Do your glycolysis. So fructose 2,6-phosphate is a positive allosteric activator. It is an activator of PFK1. It is an activator of PFK1. So basically, remember, this is ulta, like 2,6-bisphosphate is activator of PFK1. Similarly, 1,6-bisphosphate is an activator of PFK2. Okay, that's it. Then you have from 1,6-bisphosphate, there is you have glyceraldehyde 3-phosphate, and this enzyme is aldolase. No one cares, but aldolase, this enzyme. Then from glyceraldehyde 3-phosphate, 1,3-BPG. Now this sounds weird, 1,3-BPG. So something must be happening here. What is happening? Just remember that here you will have NADH. There will be a release of NADH. And another thing, see, till fructose 1,6-bisphosphate, it was 6 carbon compound. 6 carbon compound. From here you have 3 carbon compound, which basically means everything that I'm writing will be multiplied by 2. So it is one NADH. So but there are 2 glyceraldehyde 3-phosphate, so it is actually 2 NADH. Anyway, so then 1,3-BPG say 3-BPG. So something is gadbad, like how there were 2 bisphosphoglycerate, now there is only 1 phosphate. Where is this? Where did the other phosphate go? It means there was an ATP which was released. But actually it is into 2, so there are 2 ATP. Then from 3 to 2-PG, no one cares, it's just isomer kind of thing. Then from 2-PG to PEP, then from PEP to pyruvate, and pyruvate to acetyl coenzyme A. So here, what else is there? One important thing is that here this enzyme is the enolase, and this PEP to pyruvate is what? This is the substrate level phosphorylation, and the enzyme is known as pyruvate kinase. So this enzyme is known as pyruvate kinase. I always feel very confused to remember this enzyme, but sometimes you have to remember stuff because this is biochemistry. Okay, so here what we have to remember, one more thing is that from PEP to pyruvate there is a release of ATP, which is not 1 but actually into 2 ATP, because everything is into 2. And why I wrote enolase because this enzyme is inhibited by fluoride ions, which is the gray color vial in which you do the blood glucose estimation, FBS, PPBS, RBS, whatever. The blood glucose estimation happens in the fluoride vial, which contains sodium fluoride, and that inhibits enolase, because if the sugar in the blood undergoes glycolysis, then there will be a false low value of sugar. So sodium fluoride, this inhibits the enzyme enolase. What else I want to add is that from this pyruvate kinase is a substrate level phosphorylation. So what is substrate level phosphorylation? Substrate level phosphorylation basically means ki you don't need NADH, you don't need FADH, you don't need anything. You directly get ATP from the reaction itself. So from here you see you are directly getting ATP. So this is also a substrate level phosphorylation. Okay, and then okay, this enzyme is known as 1,3-BPG kinase. Why I told you about this enzyme? Because this is the only reversible kinase. Usually kinases are not reversible. That means once you add a phosphate or you omit a phosphate, it's forever, like it can't come back. So 1,3-BPG kinase is the only reversible kinase. That is the only reason I'm writing the name of the enzyme here. This is fluoride, blah blah blah. Okay, now let's be quick. Okay, but before that I have to do one more thing, that is from pyruvate to acetyl coenzyme A, what will happen? Whether ATP will be consumed or released or what it is. So basically from pyruvate to acetyl coenzyme A, remember that NADH is released. NADH, which will be multiplied by 2 into 2, everything. Okay, and from pyruvate to lactate, what happens? So pyruvate to lactate, it's just the opposite thing. One NADH is consumed from pyruvate to lactate, and the enzyme is what? Lactate dehydrogenase. And from pyruvate to acetyl coenzyme is pyruvate dehydrogenase. Okay, and from pyruvate to alanine, what is that? That is known as SGPT. Sorry, yes, SGPT or ALT. So this is ALT or SGPT, not SGOT. SGOT is different. SGOT is what? SGOT is this one, from oxaloacetate to aspartate. This is SGOT or AST. This is A. Anyway, the point is that pyruvate has these many fates. One more fate I was telling, pyruvate can also directly convert to oxaloacetate. And what is the name of that enzyme? That is pyruvate carboxylase. Pyruvate carboxylase. Okay. Pyruvate directly to oxaloacetate is pyruvate carboxylase, and here 1 ATP is consumed, which is basically into 2. If you calculate from above, it will be multiplied by 2. Anyway, how many ATP will be formed in anaerobic and aerobic glycolysis? So glycolysis, anaerobic glycolysis means it will come from glucose, pyruvate, then lactate. This is known as anaerobic glycolysis. Aerobic will be from glucose to pyruvate, that much. Let us calculate. First of all, aerobic glycolysis, because it is simple, you just have to calculate till pyruvate. So how much will come? See here, 1 ATP consumed, another ATP is consumed here, so 2 ATP is consumed. That is minus 2 because it is consumed, minus 2 ATP. Then there is NADH which is released. So 1 NADH forms how many ATP? It can get converted into 2.5 ATP. Remember, NADH gives 2.5 ATP, FADH gives 1.5 ATP. So that's it. 2 NADH are released, so that will be 2.5 into 2, that is 5 plus 5 ATP from NADH into 2. Then ATP here, 1 ATP is released, but it will all be multiplied by 2, so 2 ATP will be released. Then you have another ATP here, so again 2 ATP from PEP to this one here, 2 ATP. So total कितना हुआ? 5, 6, 7, 8, 9 minus 2. 9 minus 2, 7 ATP. This is for aerobic glycolysis. In anaerobic glycolysis, what happened? You have calculated till pyruvate. Till pyruvate it is 7. Here there is 2 ATP which is consumed. Sorry, 2 NADH which is consumed. So 1 NADH is equal to 2.5 ATP. So here everything will multiplied by 2 because you get it right. So NADH into 2, that is 2.5 into 2, that is 5 NADH or 5 ATP. 2 NADH or 5 ATP is consumed in this step. So this is still pyruvate. So if you ask about anaerobic, just take this extra step from pyruvate to lactate. There is already 7 ATP till pyruvate, and then for lactate you subtract 2 NADH which is equal to 5 ATP, that is 2. So for anaerobic glycolysis, only 2 ATP is released. So we are almost done with glycolysis. The only thing which I forgot to tell you is the rate limiting enzyme. So remember, the rate limiting enzyme of glycolysis is PFK1. This is the rate limiting enzyme. Also, if you have gluconeogenesis, you come from the reverse pathway and somehow you make glucose, then also the rate limiting enzyme will be the same step, but the enzyme name will change, right? The enzyme name will be fructose 1,6-bisphosphatase. Fructose 1,6-bisphosphatase will be the name of the reverse pathway enzyme. So remember, rate limiting enzyme of glycolysis is what? PFK1, and for gluconeogenesis is fructose 1,6-bisphosphatase. Okay, next what will we do? I really hate Krebs cycle. I don't want to do it. So let me finish this small stuff first. So this is what I'm telling. This glycogen story I already told you. From this UDP glucose is basically, there is glucose and then there is UDP which comes and gets added to it. And then finally, there is this primer called glycogenin. Okay. This is a stupid point, but since it came in one of the PYQs, I have to tell it. So glycogen is basically a primer. Okay, there is a small glycogen, small glycogen like string kind of thing. So once you have this primer, then dhire more glucose will get added to it. So the question which came was that glycogenin contains which amino acid residues? Remember, that is tyrosine. Glycogenin contains tyrosine residues. Okay, so that's it. The point is that UDP glucose, there will be glycogenin, and then the glucose will get added to the glycogen, and UDP will be cleaved away, it will go away. There will be glycogen. Done. What is the rate limiting enzyme of glycogen synthesis? This is the rate limiting enzyme. So glycogen synthase, the name of the enzyme is glycogen synthase, from UDP glucose to glycogen. That is the rate limiting enzyme. This is the rate limiting enzyme of glycogen synthesis. And what is the rate limiting enzyme of glycogenolysis? Glycogenolysis, to break down glycogen to form glucose. What is the rate limiting enzyme of that? That is also the same step, like from, not same step, but kind of like same, like the enzyme which will break down the glycogen, that will be the rate limiting enzyme. And what is that? You have to add phosphate to glycogen. Phosphate will be getting cleaved later, but first of all you have to add phosphate to the glycogen. So glycogen phosphorylase. So glycogen phosphorylase is the rate limiting enzyme for glycogenolysis. Okay, what else can I tell you? One more point I can say is that this glycogen synthase, this is also known as glycogen storage disorder you have studied. So this is known as glycogen storage disorder type 0. This is type 0. Okay. Why type 0? Because this is a very basic enzyme. Like everyone needs this enzyme. If this enzyme is not there, then the baby will die in the uterus itself. So you don't really get to see a living person with this disease. So that is why this disease is useless and it has no point. So it's just like type 0. So let the opposite one, like glycogen will get phosphorylated, glycogen phosphorylase, that must also be a glycogen storage disorder. So what is that? That glycogen phosphorylase, it breaks the alpha 1,4 bond. Basic stuff, it will break the alpha 1,4 bond by adding phosphate. And if this is defective, if glycogen phosphorylase is defective, then what will be the glycogen storage? That is type 5 and type 6. So 5 is basically muscle glycogen phosphorylase defect, and 6 is liver glycogen phosphorylase defect. And you must be knowing that this muscle ye defect is McArdle, M for muscle, M for McArdle disease, and liver, there is no mnemonic, just the ulta one. Liver is Hirsch disease. Hirsch disease is very rare, it never comes in the question, so you don't need to care. McArdle often comes. Why it comes in the question? You have to know. I'm just telling you the keywords. McArdle disease, it will write about exercise intolerance and second wind phenomenon. Mostly muscle, means muscle symptoms will be there. So this is the thing. And also listen, glycogen storage disorders are mostly seen in children, because many of these people die by the time they become adult. But in adults, the most common glycogen storage disorder is McArdle. McArdle is the most common glycogen storage disorder in adult. But overall, which is most common? And in children, which is most common? So that is, the most famous name, von Gerke disease. This is the most common overall, as well as it is the most common in children. Okay, von Gerke, if you have come there, then let us also discuss some of the peculiar points about von Gerke just to make a diagnosis. So von Gerke, remember, I'll give you only 2 words. One is hyperuricemia, uric acid, hyperuricemia. Second point is lactic acidosis. These 2 words, if you are seeing these 2 words in the question, then close your eyes and mark it as von Gerke. Other features are that you will have hypoglycemia, you will have hepatomegaly, renomegaly, but just fuck off. Forget it all. So remember these 2 words. For diagnosis, you only need one very small hint. Do not learn the entire thing. So this is how things are. Now what about Hirsch disease? In Hirsch disease, it's basically the same thing, and also it will not come in the exam, so just forget it. From here I also want to add one more thing which I am remembering, that similar features like McArdle features, same features, plus there will be hemolytic anemia. All these things will be there, plus you will have hemolytic anemia. This is known as Tarui disease. Tarui disease is also known as type 7 glycogen storage disease, and this will be seen if there is a defect in the enzyme PFK1. This is Tarui disease, and TARUI, Tarui disease, and this has features of McArdle and hemolytic anemia. Okay, hemolytic anemia से अब आते हैं कि in glycolysis, glycolysis cycle, there is hemolytic anemia with PFK1 deficiency, no doubt. With any enzyme deficiency, you can have hemolytic anemia. But tell me the most common enzyme in glycolysis whose deficiency is associated with hemolytic anemia. The most common one, give me. Very good. That is pyruvate kinase. Pyruvate kinase will be the most common which is associated with hemolytic anemia in the glycolysis. Okay. And also, since we have done type 5, 6 and 7, let us do the other types also. Type 1 to ho gaya von Gerke. See, most common and everything, this is the type 1. So where is type 2, 3 and 4? So type 2 is very interesting because type 2 is the only glycogen storage disease which is also a lysosomal storage disease. So type 2 is what? Type 2 is Pompe disease. It is both, only disease which is both lysosomal and glycosomal storage disease. And what is the enzyme defect? The enzyme defect is alpha glucosidase. Also, there is another name for it, acid maltase. Anyone can come in the option. Do not neglect, remember both the names. So this is the A. And what is the classical feature which you have to make the diagnosis? There will be cardiomyopathy or cardiomegaly or something. Some heart thing will be given, something related to heart. And what is the function of this? It will break the alpha 1,4 bond. It will break the alpha 1,4 bond. This is Pompe, type 2. Then what is type 3? Type 3 and type 4, remember, they are branching and debranching. There is a mnemonic for it, A, B, C, D. Anderson is branching enzyme defect. Cori is debranching defect. But remember, Cori is type 3, Anderson is type 4. Cori is type 3, Anderson is type 4. And to confuse you, they will also ask you, they have asked it earlier, ki what will get accumulated in Anderson, what will get accumulated in Cori. In Cori, debranching will not happen. That means you will have small pieces of like glycogen, small pieces, which will not be fully broken down. So here you will have accumulation of limit dextrins. Dextrins is basically like small few series of glucose just added together, limit dextrins. In Anderson, what will be accumulated? Amylopectin. See, this is a core chemistry thing, so I don't want to waste time in explaining what is what. Remember, in Anderson, A for A, Anderson, amylopectin, and Cori will be limit dextrin. This is PYQ, so please remember. So this is done. Then we come to that galactose stuff. From galactose, you will have galactose 1-phosphate, and from galactose 1-phosphate, you will have glucose 1-phosphate, then it can become glucose. Go to this galactose thing. Galactose to galactose 1-phosphate, it is something like phosphate has been added. So let us call this enzyme galactokinase. And from here to here, this is GALT. Galactose 1-phosphate something transferase. Okay. So remember that galactokinase deficiency, that will lead to oil drop cataract, because if galactokinase does not work, then galactose will accumulate, then it will form galactitol, and that will get deposited in the lens of the eye and lead to oil drop cataract. And this GALT is also known as uridyl transferase. See, this is how they will confuse you. GALT also, and the other name of GALT is uridyl transferase. Okay, so this GALT or uridyl transferase is the enzyme which is associated in classical galactosemia. Remember, galactosemia, when you hear of the word galactosemia, don't rush to mark galactokinase. Or don't think that uridyl transferase seems like something in the uric acid. It cannot be this. Remember, they will give you uridyl transferase in the option. If they are very generous, they will give you GALT or something. So remember, this GALT or uridyl transferase is the one which is the galactosemia. The classical galactosemia is this. Okay. Here what you will see, same. There will be oil drop cataract, but more in the galactokinase. So there will be oil drop cataract. There will be jaundice, because the liver failure will be there. Galactose is not good for any organ, remember that. And there will be seizure, because this galactose will accumulate in the brain. And there will be mental retardation. And one specific thing is that in children, GALT deficiency leads to E. coli sepsis. Not exactly children, I mean infants. In infants, E. coli sepsis. If you cannot understand what I am saying, see E. coli sepsis. This is associated with GALT or uridyl transferase. That is classical galactosemia. Okay. What else can I teach you? Oh, wow, here is another pathway. Okay, so glucose 6-phosphate gives 6-phosphogluconolactone. So remember, from here, there is actually one NADH is produced. Then from 6-gluconolactone is ribulose 5-phosphate. Here you will have another NADH which is produced. NADH. No, sorry. I did a mistake. This is NADPH. Sorry, NADPH. I'm really sorry. NADPH. And from ribulose 5-phosphate, actually, there is ribose 5-phosphate. And finally, there will be fructose 6-phosphate, which can again become glucose 6-phosphate and become glucose. But okay, the point is that I did not write ribose 5-phosphate because it's one and the same thing. Not one and the same thing, but you get it, right? There is no point in writing it, so I'm not writing it. So the thing is that there will be 2 NADPH which is produced. So this pathway is known as pentose phosphate or HMP shunt pathway, because ribulose is a pentose sugar. Ribulose or ribose, whatever it is, this is a pentose sugar, which basically means ki it is a 5-membered ring. Okay, so it has 5 carbon, so that is why this is a pentose sugar. And therefore it is known as HMP, that is hexose monophosphate pathway or pentose phosphate pathway. This pathway. Okay. What else I can tell you about it? Okay, there is an important question which comes here. This HMP pathway has two phases. There is an oxidative phase, which is also known as irreversible phase, and then there is a non-oxidative or reversible phase. So it asks which reaction is oxidative phase and which is non-oxidative. So remember, from glucose 6-phosphate to this ribulose 5-phosphate, this much is oxidative phase. Till ribulose 5-phosphate is oxidative. After ribulose 5-phosphate, everything that happens is non-oxidative. Even from ribulose 5-phosphate to ribose 5-phosphate, and then everything is non-oxidative after that. Why this HMP pathway is important? It is important because here it needs to produce NADPH. Now the question is, what will I do with NADPH? Why do I need to produce NADPH in the first place? So for that we need to know that where the NADPH is needed. So NADPH is needed for, see, this is a MCQ point. They will ask you where NADPH is needed or where it is not needed. It is needed in all except. So the point is that NADPH is needed for glutathione reductase. This is the free radical scavenger, glutathione reductase. Then you have cholesterol and fatty acid synthesis. Then you have what? Then you have NADPH oxidase. NADPH oxidase, this is also known as respiratory burst. Can you remember something from pathology? NADPH oxidase, this enzyme defect will lead to what? So NADPH oxidase is CGD. This is also a previous year question. Chronic granulomatous disease. In chronic granulomatous disease, the defect is in NADPH oxidase. By the way, respiratory burst is not merely NADPH oxidase. Myeloperoxidase is also respiratory burst. Okay, anyway, the other MCQ point about this is that in CGD or NADPH oxidase, the test is known as nitroblue tetrazolium test. Nitroblue, BLUE, blue, tetrazolium test. So what is the speciality of this test? A positive test means blue color, which is normal, and a negative test is white color, which is like it means he has this disease, NADPH defect. This is bad. Okay, this is bad. Negative test is bad. Okay, so this is my code when I like to do this, like bad or poor prognosis or something. Whenever I need to write that word, I just make this emoji, good and bad, I just smileys. So that also saves paper and time. You can also do this. So this is the thing, that a positive test in medical field, we see that positive test means this guy is having the disease, but in this case positive test, that is blue color, is normal. Okay, I'm really feeling tired now. Okay, now okay, this is gone, this is also gone. Okay, this stupid thing here, so this is nothing. This is basically, these are two isomers. When fructose 1,6-bisphosphate splits, it splits into glyceraldehyde 3-phos and dihydroxyacetone phosphate. Anyway, so this thing is what? This is actually fructose, the fructose metabolism. Fructose will form fructose 1-phosphate, then glyceraldehyde, then that will form glyceraldehyde 3-phosphate, then it will enter the Okay, so why I have written this is that because I also want to teach you, there is PYQ na, about the fructosuria and all. So from fructose to fructose 1-phosphate, what is the name of the enzyme? From fructose to fructose 1-phosphate, it is fructokinase. Like here, from glucose to glucose 6-phosphate was hexokinase or glucokinase, like that. This is fructokinase, and deficiency of this enzyme will lead to essential fructosuria. Fructose will come out in the urine. From fructose 1-phosphate to glyceraldehyde, this is not just glyceraldehyde, it forms some other compounds also, but just remember, from fructose 1-phosphate to glyceraldehyde, this enzyme is known as aldolase B. Okay, why I want you to remember this word B is because if there is a B, there is also a A. So where is A? Aldolase A is in the muscle and aldolase B is in the liver. And this liver aldolase, if this is deficient, then it will lead to hereditary fructose intolerance. Hereditary fructose intolerance. Aldolase B deficiency, that is from fructose 1-phosphate to glyceraldehyde, that will be blocked. So what are the features? In essential fructosuria, there is nothing. Okay, fructose will come out in the urine, no problem. But other than that, nothing. Okay, but in this case, aldolase B defect, that is hereditary fructose intolerance, you will have severe refractory hypoglycemia and you will have hepatomegaly and jaundice. Okay, so let us do this stupid part first. From PEP to oxaloacetate, there is this direct pathway, and that enzyme is PEP carboxykinase. PEP carboxykinase. It doesn't have much of an importance, but the only question which can come is that this enzyme, it needs GTP. You need GTP. You must have seen that GTP can be released in some cycle. There's few cycles where GTP may be released. But have you ever seen a reaction where the enzyme will need GTP? So this PEP carboxykinase needs GTP. Okay, that's done. What else can we study? Let us finish this stupid stuff first. Acetyl coenzyme A will, like 2 molecules you need, and 2 molecules will join to form acetoacetyl coenzyme A. That will become HMG coenzyme A, and blah 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 blah. So what is the enzyme? From this acetoacetyl coenzyme A to HMG coenzyme A, you are making HMG coenzyme A. So this is HMG coenzyme A synthase. So this is the rate limiting enzyme of what? Rate limiting enzyme of ketogenesis. Ketone body synthesis. Rate limiting enzyme is HMG coenzyme A synthase. From here, this is a common. Till this it is common. Then it will either go towards ketone body synthesis, or it can go towards cholesterol synthesis. So from HMG coenzyme A to then it will give acetoacetate. Acetoacetate is a beta hydroxybutyrate. Then beta hydroxybutyrate will go into the blood. Achha, this acetoacetate is the first ketone body to be formed. So acetoacetate is one of the first ketone body, or it is also known as a primary ketone body. So this is primary. Then beta hydroxybutyrate is like, it is the most common ketone body. It is the most common ketone body, and it is like highest quantity of ketone body in urine is because of this beta hydroxybutyrate. Okay, after that what will happen? You have all heard of 3 ketone bodies. So where is acetone? Acetone, wo kahan gaya? So this is formed, right? This is formed in that liver. Then this will enter into the blood. These two will enter into the blood. They are interchangeable, acetoacetate and beta hydroxybutyrate. Then beta hydroxybutyrate can turn into acetoacetate, and acetoacetate can split into acetone. Similarly, acetoacetate obviously it can split into acetone, and acetone can, like in the blood it is there. Okay, then in the lungs also it will go. Then when you breathe out in expiration, acetone can come out in your breath when you expire, and that acetone has this fruity odor. That is why you say that in ketoacidosis or something there will be a fruity odor in the breath. That is due to acetone. Fruity odor is due to acetone, because acetone is volatile. Now, since we are doing this anyway, so let us know a few other small things. So remember, this conversion from this acetoacetyl to HMG coenzyme A is HMG coenzyme A synthase. Then from HMG coenzyme A to acetoacetate, this is HMG coenzyme A lyase. This is HMG coenzyme A lyase. Also, there is another enzyme called thiolase. In ketone body synthesis, you have need to remember 2 confusing enzymes. One is thiolase, another one is thiophorase. So thiolase is actually 2 acetyl coenzyme A will combine to form acetoacetyl coenzyme A. So you can see that it should be acetyl coenzyme A, acetyl coenzyme A. Where is the other coenzyme A? If 2 will mix to form 1, then it see, there are 2 acetyl, but there is only 1 coenzyme A. So some reaction is happening here. So this enzyme which converts to acetyl coenzyme to acetoacetyl coenzyme A, that is thiolase, L-A-S-E, thiolase. And so what is the thiophorase enzyme? Thiophorase enzyme is like from acetoacetate. From acetoacetate, it will form acetoacetyl coenzyme A, and finally it will become acetyl 2 into acetyl coenzyme A. So this how will I write it? Let me do this. Acetoacetate to acetoacetyl coenzyme A. This is thiophorase, and here actually there is a coupling. Coupling means what? There is a succinyl coenzyme A. It will become succinate. Okay, succinyl coenzyme A will give its coenzyme A to acetoacetate. That will become acetoacetyl coenzyme A, and because of losing the coenzyme A, it will become only succinate. Okay, and this enzyme is the thiophorase. So the conclusion is that thiolase is the enzyme involved in ketone body synthesis, but thiophorase is the enzyme which is involved in ketone body breakdown. Isn't it? Got it? What is the difference between ketone body synthesis and breakdown? Remember, ketone body synthesis occurs only in the liver. Ketone body breakdown will occur everywhere except liver. Why? Because liver lacks the enzyme thiophorease. It is not present in the liver. That is why liver is not involved in ketone body breakdown. Now let us know some interesting things. What are common? What is common between ketone body synthesis and breakdown? First thing, both are catabolic. Both are catabolic. Second thing, both of them occur in mitochondria. Third thing, both are activated by glucagon. I don't have time to explain you, so just remember. Both are catabolic, both happen in mitochondria, both are activated by glucagon. Done. Okay, next come to the other thing of HMG coenzyme A. Now where it will go? HMG coenzyme A will become mevalonate, and from mevalonate there is actually another compound, farnesyl pyrophosphate. Should I write it? Okay, let me write it. Farnesyl pyrophosphate. From here it will form what is that stupid compound? Squalene. From squalene you will go to lanosterol, and over time ergosterol, blah 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 blah, and then you will have cholesterol. This is a previous year question. How many carbons are there in cholesterol? 27 carbon. Okay, why I wrote about lanosterol? Because lanosterol is the first cyclic compound in the cholesterol synthesis pathway. First cyclic compound is lanosterol. What else? Why I wrote about farnesyl pyrophosphate? Because farnesyl pyrophosphate, after that it becomes coenzyme Q. And where is coenzyme Q? Coenzyme Q enters the ETC. So that is the importance of farnesyl pyrophosphate. Achha, what is the rate limiting enzyme of cholesterol synthesis? HMG coenzyme A reductase. This thing from HMG mevalonate, this is HMG coenzyme A reductase. This is the rate limiting enzyme of cholesterol synthesis. And statins inhibit this enzyme. This is also a previous year question. What enzyme is inhibited by statin? HMG coenzyme A reductase. Okay, this stupid thing, odd chain fatty acid, isoleucine, valine, methionine, threonine and pyrimidines, they become propionyl coenzyme A, then methyl malonyl coenzyme A, and finally succinyl coenzyme A. So what is there? So listen, this isoleucine, valine, methionine and threonine, remember they are both keto and glucogenic amino acids. This is one PY important question. Both gluco and ketogenic amino acids. What else is there? Another thing is that here you need B12 from methyl malonyl coenzyme A to succinyl coenzyme A. B12. Okay, B12. What else I have to say? Acha, B12. From B12 I remember there are some other things I totally forgot to tell you. So listen, this pyruvate carboxylase is there na here. Pyruvate to oxalate, pyruvate carboxylase. Here the enzyme coenzyme is biotin. Biotin is what? B7. B7 biotin, okay, pyruvate to oxaloacetate pyruvate carboxylase, and this lactate, this LDH has a B3 cofactor, and ALTA, SGPT, whatever, this one has B6, and what about PDH? PDH has 5 cofactors. PDH has how many cofactors? 5 cofactors, 3 enzymes and 5 cofactors. What are the 3 enzymes? First, the first enzyme is pyruvate dehydrogenase, okay, and the second enzyme is dihydrolipoyl transacetylase. Third is dihydrolipoyl dehydrogenase. So, do I need to write it? Dihydrolipoyl transacetylase. Then dihydrolipoyl dehydrogenase. 1, 2, 3. So remember, and there are 5 cofactors. The mnemonic is 1, 2, 3, 4, 5, where 4 is not 4. 4 is lipoic acid. Okay, so 1, 2, 3, lipoic acid, 5. Basically B1, B2, B3, lipoic acid, and B5. So remember this PDH, this coenzyme is, cofactor is B1. This is B1, this is 4, 5, and this is 2, 3. Not 4, 5, I told you, no, 4 is not 4. 4 is lipoic acid actually. So this is, B1 is thymine, not exactly thymine, it will be TPP, thymine pyrophosphate. Then lipoic acid is lipoic acid, and 5 is what? 5 is pantothenic acid. Pantothenic acid is not pantothenic acid. Pantothenic acid, the form, one of the nice important forms of pantothenic acid is coenzyme A. So this is the basic thing na. Everyone should know कि pantothenic acid or B5 is coenzyme A. It is not exactly coenzyme A. Just remember that this is a form of B5. Coenzyme is a form of B5. Remember, this is a very basic thing. Anyway, that's it. Then this dehydrogenase is 2 and 3. What is B2? B2 is what? B2 is riboflavin, but not exactly riboflavin. The thing will be FAD. Flavin adenine dinucleotide. FAD, flavin. Then 3 is what? 3 is niacin, but it's not niacin exactly. It will be the other form of niacin, NAD, niacinamide adenine dinucleotide. Okay, that is PDH. Now in the exam they might ask you all of the following are cofactors of PDH, except they will give from this 5 options they will give 3 options, and one will be odd one out, some other random option you will mark it. Sometimes They will confuse you by putting all from these 5 options. All the options will be from this 5. Like they can give the options as B1, B2, B3, B4. Then what you will write the answer? Then you will pick the most appropriate option. Which is the most appropriate option? Since the first coenzyme of PDH is PDH, the most appropriate option will be B1. If all will be there in the option and you have to select only one, then the answer will be B1. Okay, before this TCA, let me just finish this. Oxaloacetate to aspartate, this enzyme is SGOT or EST. Here one NH2 will be added, then it will become aspartate. Aspartate and citrulline, they mix to form arginosuccinate, and arginosuccinate will form arginine. How? This will go to fumarate. Okay, then arginine, ornithine. Ornithine plus carbamoyl phosphate will form citrulline. Carbamoyl phosphate is formed from NH3 plus CO2. So what is the question here? Question here is from ornithine to citrulline. Is ornithine trans carbamoylase? OTC is the enzyme, and OTC defect is the most common deficiency. And this is known as which type? Type 2 hyperammonemia. OTC defect is known as type 2 hyperammonemia. Okay, then what is type 1 hyperammonemia? Type 1 hyperammonemia is CPS1. CPS, what is the full form of CPS? Carbamoyl phosphate synthase. Carbamoyl phosphate is being synthesized, so carbamoyl phosphate synthase 1. This is the type 1 hyperammonemia. So now another question. This CPS1, this is the rate limiting enzyme of what is the cycle? Urea cycle, rate limiting enzyme of urea cycle. Now another question, if there is a CPS1, then there is also a CPS2. So what is CPS2? CPS2 is an enzyme which is involved in the pyrimidine synthesis. So this is another thing you should know, CPS1 and 2. So 1 is here in urea cycle and 2 is in pyrimidine synthesis. What other difference? CPS1 is present, this enzyme is present in the mitochondria. CPS2 is present in the cytoplasm. Got it? In exam, usually they will ask you from OTC deficiency, hyperammonemia. So usually this OTC deficiency, this will present with something like a child will be there, and the child will present with hypoglycemia, failure to thrive, that kind of thing. And there will be a history of high protein diet. Okay, history of protein rich diet will be there in that child, and from there like it's a kind of a signal key. Most probably this is OTC deficiency. Okay, now we have to, one more thing, right? Urea. See from where urea is there. Actually arginine, if you add water here, then urea and citrulline will be formed, and this urea will obviously that will come out through the kidneys. So urea, this is a very common question. Urea, what is the formula for urea? NH2CONH2. This is the formula. So where it comes? One NH2 comes from ammonia, one comes from CO2, atmospheric CO2, another NH2 comes from aspartate. So remember, aspartate is the source of one of the nitrogen of the NH2. In pyrimidines are also nitrogenous bases, right? CPS2, which is involved in pyrimidine synthesis. So remember, in pyrimidine also it's a nitrogenous base, so there is nitrogen and carbon. So in pyrimidines, the carbon is from CO2 and the nitrogen is from glutamine. The nitrogen is from glutamine in pyrimidine. Done. Before I come to the elephant in the room, what did I miss out? Okay, I missed out on this, that PDH is link reaction. PDH is also known as link reaction because it links glycolysis with Krebs cycle. Okay, now elephant in the room, oxaloacetate plus will basically citric acid cycle or TCA cycle. OA plus acetyl coenzyme A will form citrate, and citrate is what? Citrate is a 6 carbon compound. Because acetyl coenzyme is 2 carbon, oxaloacetate is 4 carbon, so this is 6 carbon. Then from citrate it will form isocitrate. What is the name of the enzyme? The enzyme is known as aconitase. Aconitase, and it is inhibited by a substance called fluoroacetate. Then isocitrate, from isocitrate you will have alpha ketoglutarate, and what will be released here? You will have NADH. NADH will be released, and the name of the enzyme will be isocitrate dehydrogenase. Then from alpha ketoglutarate you will have succinyl coenzyme A. The name of the enzyme is very famous, alpha ketoglutarate dehydrogenase. Is anything released here? Yes. Again there will be an NADH which is released. Then succinyl coenzyme A is this is 6, and isocitrate is also 6. This is also 6 because isocitrate, isomers 6. Alpha ketoglutarate is 5 carbon and succinyl is 4 carbon. Succinate is obviously again it's the same 4 carbon. So succinyl coenzyme A to succinate, this is a substrate level phosphorylation. Why? Because it will lead to release of a GTP. GTP, remember what is released? GTP. Now, succinate will form fumarate. So here, what is the name of the enzyme? SDH, another famous enzyme, succinate dehydrogenase. Okay, and what else will be released? You will have FADH2, FADH2. What else? Okay, why I said SDH is famous? Where did you hear it? You have heard it in ETC, electron transport chain. Which complex is SDH? SDH is also the complex 2 of ETC. What inhibits SDH? Malonate. Malonate inhibits SDH. So from here, what did you understand? You understand that if malonate is more, then TCA cycle will be inhibited. Or it can be written differently. Malonyl coenzyme will inhibit TCA cycle, but methyl malonyl coenzyme will activate TCA cycle. Because if there is more malonyl coenzyme, then more product, there will be a tendency to form more product to get rid of the methyl malonyl coenzyme. So methyl malonyl coenzyme activates TCA cycle, whereas malonyl coenzyme or malonate inhibits TCA cycle by inhibiting SDH. Okay. Now fumarate to malate, nothing, it's the same like another 4-carbon compound. Oxaloacetate is another 4-carbon compound, but from malate to oxaloacetate there is an NADH which is released. What else do I need to say? This oxaloacetate is also, this is a MCQ, which is the first substrate of TCA cycle. Oxaloacetate is the first substrate of TCA cycle. Now let us go to the energetics. Energetics basically means like aerobic glycolysis. Okay, not even aerobic glycolysis. What will I tell this? Like complete oxidation of glucose. You must have heard complete oxidation of glucose in the presence of oxygen will give 32 ATP. You have heard this sentence. How 32 ATP is happening? So for that we have to add all the steps. Before adding all the steps, let us at least add the TCA cycle, because we just studied TCA cycle, right? We'll do this cycle. What is happening here? NADH, NADH, NADH. There are 3 NADH, 1 FADH2 and 1 GTP. Okay, into 2, because 1 glucose into, this will split, blah blah blah, so everything will be multiplied by 2. NADH, 1 FADH2 and 1 GTP. 3 NADH is equal to 3 into 2.5. FADH2 is equal to 1.5. Okay, so 1 into 1.5, 1 GTP is 1 ATP. Okay, and everything will multiplied into 2. So this is equal to 7.5, 1.5, 1. How much? 7.5 plus 1.5, 9 plus 1 is 10. 10 into 2. Everything will multiplied into 2 for 1 glucose. If 1 glucose enters, then this will be into 2. Into 2, that is equal to 20 ATP only for this particular cycle. For TCA. In TCA, 20 ATP is released and nothing is consumed. There is no ATP or anything consumed here. That is it. Now, we calculated that till pyruvate, we got 7 ATP. Didn't we? Till pyruvate, what was the calculation? 1 ATP plus 1 ATP. 2 ATP consumed, which is basically minus 2. Then, yes. So, minus 2 and all this will be multiplied by 2. So, basically, you will release 2 NADH. That is 2 into 2.5, that is 5. And 2 ATP. Another 2 ATP. So, that was equal to how much? Not even 2, sorry. 2. This is another 2. 4 plus 4. 9 minus 2, 7 ATP. Till this much, 7 ATP. So, till this much 7 ATP, this is 20, and this here there will be a release of 1 NADH, 1 which will be multiplied by 2 into 2, this NADH, link reaction, link, here 1 NADH, but it will be into 2 because everything from here onwards is multiplied by 2, NADH into 2, that is 2.5 into 2 is 5. Now add it, 7 plus 5 is 12, 12 plus 20 is 32. Did you get the answer? 32. 7 plus 5 plus 20. This is the total number of ATP if one molecule of glucose completes the TCA cycle. One more fact I forgot to tell you, which is the rate limiting enzyme of TCA cycle. Very unexpected enzyme, isocitrate dehydrogenase. This one. From isocitrate to alpha ketoglutarate. Isocitrate dehydrogenase. This is the rate limiting enzyme of Krebs cycle. Done? Done. So let us also do the energetics of beta oxidation of fatty acids. So that we had seen from acetyl coenzyme, there was malonyl coenzyme, from there was fatty acid. So remember fatty acid oxidation, that is beta oxidation. is two types. One is even chain fatty acid and odd chain, it is separate. For even chain fatty acids we had seen it, for odd chain fatty acid we had seen it here, like odd chain fatty acids and all those amino acids, they were becoming propionyl coenzyme A, then they were becoming propionyl coenzyme A, then it will become methyl manolin coenzyme A, then they were becoming succinyl coenzyme A and entering the TCA cycle. So that is very different. But what will come here is even chain fatty acids. We will learn about that. Even chain fatty acid will also enter the TCA cycle at this level, acetyl coenzyme A. But how does it happen? So let us take an example of palmitate, like 16 carbon palmitic acid, that is a fatty acid. So it will be split into 2 fatty acids. So 16 carbon means it will give rise to 8 acetyl coenzyme A, and this 8 acetyl coenzyme A will enter TCA cycle. Now this palmitic acid, when it is getting oxidized, first of all 2 ATP will be consumed to convert the fatty acid to fatty acyl coenzyme. You don't need to know that much like how it is happening or blah blah blah. Just remember that any fatty acid will enter in the first step. First minus 2 ATP will happen to activate the fatty acid into fatty acyl coenzyme. After that you have to separate that thing into acetyl coenzyme, like 2 carbon, 2 carbon, 2 carbon. For 16 carbon here you have to make 8 fragments. 8 fragments means how many cuts are there in between? 7 cuts. Like 8 fragments, 3, 4, 5, 6, 7. So 1, 2, 3, 4, 5, 6, 7, 8. Like 7 cuts are needed to make to make 8 pieces, right? So 7 cuts means 7 times energy will be released here. So here every time one cut is made, you will have 7 FADH2 and 7 NADH. Every time you cut, you will get 7 FADH2 and 7 NADH, and in the very beginning 2 ATP will be consumed. Means if it is 16 carbon, 18 carbon, 20 carbon, 22 carbon, anything, 2 ATP will be minus. That is fixed. Not that if it is 32 carbon then 16 into 2, 32, so 4 ATP, not like that. Minus 2 ATP is fixed. So basically for this palmitic acid, what is going to happen? Let's calculate. 7 FADH2 is 1.5 na. So 7 into 1.5 is how much? I don't want to calculate this. 90, 105, 10.5. Okay, 10.5, right? Hopefully. Then you have 7 NADH. NADH is 2.5. 7 into 2.5 is how much? How boring. 17.5. 17.5, right? 17.5. This much. So total how much? 17.5 plus 10.5 is 28. 28 minus this, you added 28 minus this 2 ATP will be minus. So 28 minus 2 is 26 ATP. 26 ATP, but this 8 acetyl coenzyme will enter TCA cycle, and every time one acetyl coenzyme is entering, it will give rise to 10 ATP, which we had done it here, like 10, but for every molecule of glucose there will be 2 acetyl coenzymes, so 10 into 220, but basically for 1 acetyl coenzyme it was 10. So for 8 acetyl coenzyme it will be 8 into 10, which is 80. So the most common fatty acid, that is for palmitic acid, it will be 8 times 80 plus 26 is equal to 106. For palmitic acid, 106. So basically for every fatty acid it will be different. The calculation will be different. So thank you. That's it for today. I'm very tired.